Okay, shall we get started? Keratinocyte and trichocyte.
Yes.
Okay.
Good.
Hi, I'm Jeff. Okay, I'm a professor over the Department of Life Science, National Taiwan University. I'm very happy to be here for today. Today, our subject will be going to deal with the cellular signaling. Let's first define what is the cell signaling. Well, cell signaling is how cells sense and respond to actual cellular signal. Okay, and for this actual cellular signal, actually it can be subdivided into endogenous signal and exogenous signal. Normally, how cells respond to the outside environment. There are two types of the signal. Number one is a chemical signal. Number two is a physical or mechanical signal. Okay, and can anybody tell me what do you know about a chemical signaling? Give us an example of a chemical signaling that can trigger the cellular signaling response. Anybody? What is a signal that can trigger the cell signaling? You know. Hey, everybody, wake up. What do you know about the signal? What type of signal that can trigger the cell response? Okay, I saw there's a student with a white lung tissue and somehow and sit in the first row, with your notepad on your desk. Hello? Nobody? Okay, let's go back to the NTU side. That's easier to deal with. Okay, you. Okay. Yeah, you. That's you. Yeah, that's right, exactly. What do you know what type of signal we have can trigger the cell response? Put on your microphone. We heard that every day, right? And we have a lots kind of what signal that can trigger the cellular response in your body.
Electronic.
Serotonin. Oh, good. What is serotonin? Serotonin is a neurotransmitter, right?
Yeah.
Yeah, that can trigger the neuronal response. What else? We have lots kind of hormone, right? Okay. For example, insulin. Well, insulin have insulin-dependent response, etc. Okay. But anyway, and why cell need this signal to do that? And in fact, those kind of what they call this kind of signal normally we call it first messengers. First messengers. That's a messenger role that can trigger the response. And this first messenger normally will be extracellularly. Okay, you can produce in body, like for example the glands or that kind of hormonal gland. Okay, and that can secreting hormone circulating in the blood vessel, and that's in endocrine signaling. And also cell itself can screen some signal as well, cytokines, right? And cytokine can trigger the cellular response. But well, once you have the first messengers, what's next? This first messenger need to be able to trigger the cell response, either by touching the cell surface receptors, okay, or it can penetrate through the membrane, go into looking for intracellular receptors. And once this receptor got activated, it will trigger cellular response in the cytosol, okay? And normally, cell signaling have two types of response. One is transient, the other one is permanent. Okay, when you have the transient response, normally the response occur in cytosol. Okay, and for that kind of response to occur in cytosol, normally you have the activation of enzymes, for example, a kinase. Okay, a kinase is the enzyme that can put phosphate on the protein. And normally, is an enzyme got phosphorylated, it can either activate the response or inhibit the response. Okay, and modulation of this kind of kinase enzymes that can trigger the downstream pathway. And the other type of the first messenger, the response is permanent. And that kind of permanent response normally it trigger the expression of the gene. Okay, it go into the nucleus and trigger the expression of particular gene, for example, a transcription factors. And once you have a transcription factor produced, it's going to cause what? Cause a change in other gene expressions. And that way, you can somehow change the cell shape of other things, that produce a permanent response. Okay, and it doesn't matter whether it's a transient or permanent. There's a very, very important criteria for the cell signaling, that is the specificity. Okay, the student on the right here, can you explain to me what is the specificity? And why do we need specificity?
Because the signal the specificity allow the signal to trigger only some type of cells or some type of gene expressions.
Very good. Okay. Normally, specificity is key for cell signaling because we all know that there's a signal to noise ratio. It doesn't matter what type of signal. It always has really actual signaling and also have a background noise. So if the specificity of the first messenger is not high enough, you're going to have the signal and noise together. And in that way, how can the cell sense what is the signal, what is the noise? And what is the best way to control the specificity? What is the best way to control the specificity? Well, normally, when we call about first messenger, the amount of first messenger is very, very low. For example, the hormone circulating in the blood vessel, the hormonal level concentration is quite low. And why we need to keep those hormonal concentrations at low level? Because the lower the amount, the higher the specificity, right? That's one key of it. So normally we want to try to measure the first messenger. Actually, it's pretty difficult. Okay, in a specific method, in order to measure the first messenger level, for example, hormone, you can use RA or EIA ELISA to try to detect the hormone level. Okay, and the level is very low. And the reason I keep it low is that we want to provide specificity. Okay, and in addition to the concentration of the first messenger, those first messenger always bind to its respective receptor with a high affinity. Okay, for example, one type of first messenger can only bind to a specific type of the receptor. Can anybody here give me the example of what do you know about the messenger and the messenger here, we call it a ligand and receptor pair. You know about the ligand and receptor pair? Any student there and QSI? No, but give us an example about ligand and receptor, Hello? Wave your hand if you hear me. Oh yeah, the one, looking in the back. The second one, the girl. Anybody there? What do you know about ligand and receptor pair?
Is it like an example or what it is?
Yeah, example. What do you know about the hormone?
I can think of like G proteins and G proteins.
Exactly, very good. Thank you very much. That's what I'm going to get into. Okay. The history of the cell signaling is for a long, long time. Long before we know about the term cell signaling. Before the cell signaling term comes up, we always talk about hormonal action, the action of the hormones. Okay? And there's a lot of the good stories and good scientific findings about cell signaling have been acknowledged to get a Nobel Prize. And one of the first, this type of story is the G-protein coupled receptor. You see it in 1971, okay, Dr. Sutherland, okay, the faculty over the Vanderbilt University over US, he discovered a very, very important hormonal action that is a very common hormone, epinephrine, that is adrenaline. Anybody here can tell me what is the action of adrenaline? Screen is Tsukubasai? What do you know about adrenaline? Okay, Tsukuba student, wave your hand. Nobody there? Okay, adrenaline actually is a quite common hormone, and everybody know that, right? When you see the NBA play off, okay, now NBA is playing off, right? They get into the Western and Eastern final. And they always pump their chest. What is that for? Pump up the adrenaline. When your adrenaline comes up, your muscle gets strength, and you can jump farther, and you can jump higher, right? Less adrenaline. And even when you just step under the desk, okay, everybody raise their head, okay? And how does some sort of adrenaline? It's a fight and flight response, okay? So the action got to be very, very quick in a very specific way. And how can adrenaline or the other name is epinephrine do? Actually, it activated on a very important receptor, okay? That's the G-protein coupled receptor, okay? And this G-protein coupled receptor, what do you call it G-protein? Because it is mediated by a very small protein that is called G-protein, GTPase activating protein, G-protein, okay? And somehow it's a heterotrimeric G-protein, and for that, it will activate downstream enzyme called adenylate cyclase, okay? The adenylate cyclase is a quite common cyclase that can produce a very important, that is the second term, is called second messenger, okay? Second messenger is try to respond to the stimulus from the receptor and produce in the cytosol. In this case, will be the cyclic AMP, okay? And the cyclic AMP actually is the second messenger produced in the cell, And once it produced in the cell, it can bind to the cyclic AMP dependent molecules. In this case, we can call it a kinase called cyclic AMP dependent kinases. And the abbreviation is pKa, okay? And this is one of the first kinase been discovered. pKa, protein kinase A, okay? And once this protein kinase A got activated, it can the phosphorylated other enzyme required for a lot of action. For example, pump up your muscle. You can jump farther or can fly away. And this type of response cannot be last longer, okay? You don't want to be excited all the time, right? You need a strength when you need it. And once it's done, this cyclic AMP should give away, degrade it away, and they stop the response. Otherwise, you're getting crazy. So when you're looking at this kind of signaling, actually, there are several things you got to be very careful about it, about the cell signaling. Number one, ligand first messenger in receptor specificity. Okay, and once receptor got activated, you need to transmit a transmitted signal into the cytosol. And however, there's one thing got to be done in the cytosol. That is called amplification. You need to amplify the signal. Remember that the number of the first messenger is very, very low. Okay, and one ligand can only bind to one receptor. That's 1-to-1. However, if the receptor can only activate one G protein or produce one cyclic AMP, the response will not be strong enough. So how can the signal get amplified? Well, this G protein can activate adenine cyclase, and this adenine cyclase can produce many cyclic AMP. Okay, and once you have many cyclic AMP, you can activate many protein kinase A, and once you have many protein kinase A activated, you can trigger the response in a strong enough way to do it. Okay, so specificity and amplification is one of the key for successfulness of the cell signaling, and this has been discovered back to 1971, long time ago. Okay, any questions so far? Okay, so remember that, okay, you need to have a first messenger activate a receptor, and this receptor activate G protein, that's a transducer, okay? And once this transducer got activated, you activate downstream effector, okay? And this effector will then produce cyclic AMP, okay? And then downstream activate something, another type of effector, or postal activation of cyclic AMP, okay? So, and this individual step can have an amplification occur, and that to strengthen the signal. And also, when we have transducer, we have cyclic AMP, it also has a way to turn off the signal. How can that be done? Okay, once the G protein got activated adenine cyclase, how can that be done? Because originally this G protein is a heterodimer G protein. And once it got activated, it would dissociate between GS and G beta gamma. Okay, the GS is the stimulatory G protein, can activate the adenine cyclase, and G beta gamma actually is some sort of the inhibitory thing. Okay, but what we call it G protein because it relies on the GTPase, okay, activity. Once you have the GTP become transformed into the GTP bound form, the G protein become inactivated. Okay, once it become inactivated, G protein action got turned off, so they will no longer activate the adenine cyclase. And once this cyclic AMP somehow got degraded, you will no longer be able to activate the protein kinase A, and then the entire signal got turned off. Okay, so basically, specificity, amplification, and turning off degradation of the signal is the key to contain the really proper amount of the response to the first messenger. Okay, and we already taking the adenine cyclase as a very good example for serotonin signaling. Now, in 1992, actually, that is 1971. At this time, they only know that epinephrine can activate the adenine cyclase, produce cyclic AMP and some actions, but they don't know about the pKa, okay, kinases. 1971, then back to 1992, long time later, right? So, Dr. Fisher and Dr. Crabb over University of Washington, okay, and they discover the action of the phosphorylation protein, reversible phosphorylation, okay. And what we call reversible protein phosphorylation is so important. That is in the case for the protein kinase A. Protein kinase A phosphorylated molecule, right? And you put a phosphate group on, and this molecule has activity. But you got to turn off the signal. And how to turn off the signal? Well, dephosphorylate it, right? So, and this kind of dephosphorylation is a reversible process. That means what? You can turn it on and off at your will if you properly control. So, by that way, people discover that reversible phosphorylation actually is one of the key mechanisms to control the cell signaling. And we talked about G protein. Okay, the G protein is not discovered until 1994, okay, by Dr. Gilman and Dr. Robel, okay. And they discovered G protein, and this is a very, very important discovery because it is a really widely used for our body system. And as far as we know that 100% of the protein are G protein coupled receptors, okay. And later on, this G protein complexes have been found in a variety of signaling contexts, including olfactory sense, okay, the receptor in your nose, olfactory bulb. And actually, I cannot remember this year two. Somehow, recently, there's another Nobel winning story. That is that they discovered an olfactory receptor. And one olfactory receptor can only sense one first messenger chemical signal, olfactory material, okay? And then in your nose, you can discover thousands of the olfactory receptor can sense different kind of smell or odor. And they are all G-protein coupled receptor. Okay. And this is in the receptor type level. And the other type of level is that the signal that can penetrate through the membrane. You don't need receptor on the membrane. Okay. And for example, what type of signal is that? Carbon dioxide, okay, and nitric oxide. Here, anybody here know nitric oxide? People in the school outside, do you know nitric oxide, NO? Nobody? NO, very small, very simple molecule. Nitric oxide. Nitric oxide is an active ingredient in Viagra. I'm sorry, not really, okay, different thing. Nitric oxide is very important signaling first messenger. Actually, okay, it can easily penetrate through membrane because it's a small little molecule can penetrate through membrane. And what it is for? Once this nitric oxide go into the membrane, well, it can trigger the muscle relaxation. Okay, and how can it cause the muscle relaxation? Well, the point is that it activate cyclic GMP phosphatidylserase. Okay, that is the one can somehow degrading cyclic GMP. Okay, and the cyclic GMP is another second messenger that is important for trigger the muscle contraction. And once we degrade cyclic GMP, somehow this muscle will become relaxed. Okay, and they discovered this, and actually this has been applied to synthesis of the discovery of the very important drug, Viagra. Okay. So basically, so far we know about the extracellular signaling, including the physical factor and chemical signaling. People know a lot about chemical signaling, but how about the physical factors? How can the cell sense the physical factor? And this field actually is not well studied, but people begin to have a very great interest in that. For example, if you squeeze a cell, how can the cell know? That's the mechanical stress, right? Okay, and cells, sure, definitely, they can tolerate some sort of squeezing or stretching. And how to, if this cell squeezing and stretching, somehow will change the cellular, induce the cellular response. For example, shear stress. Okay, your blood, circulated in your blood vessel, right? And inside the blood vessel, in the most interior part, there's a layer of called endothelial cells. Okay, endothelial cell blood vessel, and the blood circulates very fast in the blood vessel, and that means what? This endothelial cell constantly face the flow of the blood, and this is because of the physical. Okay, and some sort this kind of physical state can either change or some sort of response, and the same thing happen in during different kind of cellular environment as well. And that's why you always have hair. You have hair, right? Not only your hair, but also inside the cell you have cilia. There is a cilia, and this cilia actually can sense the fluid. The direction of fluid, circulates through it. Okay, and once you have a the cilia beating, actually it can change the cell interior gene expression response. Okay, for example, people here actually, if you cut yourself into half, you can become equally, similar to half. But in your body, actually, the internal organ, there are what they call bilateral asymmetry, because some of the organ in the left side, some of the organ on the right side, right? And how do you know, how do the organ know which side to situate at? It is very early during development. There is a very transient organ called nodal. Okay, a nodal contains a cilia, and the cilia can control by the fluid movement, and they control either it should express the left side gene or express the right side gene. And once you know have the expression of left or right side gene, that will subdivide your body into the left side looks phenotype and right side look phenotype. Okay, and that is the stress response. Okay, but somehow stress mechanical response is not easy to study. So that's why early scientists always look at the chemical signal because they have the ability to find target to measure. And physical response is not easy to study, but we gradually know something about it. Okay. Okay, and the other thing is how this is a very long story. Actually, I get this from a very old molecular biology book because those ligands need to reach the cell, okay, and by several ways, including circulation in the blood or the gas transfusion, okay. Then it doesn't matter what, you got to be able to reach the cell in touch. And you have the endocrine system, you also have the paracrine system, and you also have the paracrine system, okay? Okay, either you can activate on a distant cell, or activate a neighboring cell, or activate itself, okay? In this case, this is a hormone, okay? And also paracrine and endocrine will be cytokine. So we already talked about this. So you have different ways to reach a sensitive ligand, membrane receptor, intracellular receptor, and receptor in the nucleus. So 3 different layers: membrane, cytosol, and the nucleus. And how do the cell respond to the ligand? You can activate the receptor, signal relayed by the second messenger, you modulate effector activity, and then change the cell response. And this cell response has many types: cell growth, shape, migration, cell death, okay, and others. And there are different types of first messenger. We know mostly are secretory form, okay, including hormone in the circulation, neurotransmitter in the synaptic cleft, and also the local mediator in the extracellular fluid. And we have different types of the first messenger, including amino acids, water-soluble metabolites, lipid, and peptides, okay? And different type of first messenger actually have very specific actions in the circulation. And this table, you can look at that, pretty much tell you many of them, including we already talked about adrenaline, noradrenaline, insulin, glucagon, blah, blah, blah. And also, you also have neurotransmitters, mostly have the tyrosine or tyrosine derived. Acetylcholine, the ACH is very well-known neurotransmitters. Adrenaline, we already talked about it. Dopamine, serotonin, we already talked about that as well. And also, the amino acid have the neurotransmitter, glutamate, and glycine as well. So, chemical signal actually is one of the good examples, even before we know about the term cell signaling. That's neuronal transmission. Basically, there are 2 different steps that is critical for the neuronal transmission. Number 1, the synthesis of the neurotransmitter, right? This neurotransmitter got to be synthesized. If you have a problem with synthesizing neurotransmitter, for example, dopamine, okay, now you're going to the normal membrane loss problem. And once this neurotransmitter got synthesized, you need to restore in the presynaptic neuron until you need it, okay? And those stored neurotransmitter vesicles, once they need it, it got to be transported to the membrane of the presynaptic cleft, okay, to be ready for secretion. That is a term called exocytosis into the synaptic cleft. And how can the vesicle transport to the membrane? And how can the vesicle fuse with the membrane then become exocytic? Okay, this is heavily studied. Okay, and then once it got secreted into the synaptic cleft, actually, you got to be able to reach the postsynaptic neuron by different way. And this is what we call it ligand-gated ion channels or others. Okay? And once the ligand binds to ligand in the ion channel, this channel will change its opening, either open or close. Okay? And once they open or close, will allow the ion go able to activate the postsynaptic neuron, okay? And then you can fire the neuron by the chemical stimulation, okay? And it doesn't matter what type of, either it's a chemical stimulation or neurotransmitter activation. They also follow the rule what I just described about the cellular signaling. That is that neurotransmitter has produced a very low amount, okay, and you can, this low amount of neurotransmitter can have high specificity to bind to its receptor. In this case, can be ion channel, okay, and once the ion channel open, it need to have be amplified because once the ion channel open, it will allow many ions to go into the cell. Okay, that's the amplification step. Okay, and once this, ion go into the postsynaptic neuron, you can either activate action potential, and this action potential can be propagated. Actually, this is another sort of amplification. Okay, and then fire the entire neuron. Okay, so specificity and the amplification is a key for cellular signaling. And how about local mediators? Okay, and one of the best examples of a local mediator is the nerve growth factors. Okay, and it's one of the classical example that people can have a gel-like boxes here. Okay, and then you have culture this sympathetic neuron on the middle chamber, and then put a lot of the side NGF on the right side chamber, and then this sympathetic neurons can go pass through the gel and then go into the right chamber. Okay, that means what? This neuron can sense the stimulation from the NGF, and the NGF can attract it or stimulate the growth of the neuron, and also the stimulate migration in the neuron. Okay, and this is a very classical example for the local mediator trigger cellular response. Another thing for the wound healing. Okay, once you have the cut on your epithelial cell, well, the first thing you want to close the wound to stop the bleeding, right? So somehow this clotted platelet cell was secreting platelet white growth factor (PDGF). Okay, and the PDGF got to be able to do two things. What two things? Number one, need to attract cell to migrate to the wound, right? Otherwise, the wound cannot be healed. Number two, if this cell number is not enough, you got to be able to trigger the cell proliferation in order to get enough cell to close up the wound, the gap of the wound. Okay, so somehow this PDGF serve a role to do these two things: triggering cell proliferation and triggering cell migration. And a lot of them activating through the G-protein coupled receptor. I already told you about that, the structure for the G-protein coupled receptor, but I haven't told you about what is the structure of it. The one of the characteristics for G-protein coupled receptor, you can see that is a 7 transmembrane protein. Okay, 1, 2, 3, 4, 5, 6, 7. If you follow the protein, membrane protein, pass through the membrane for 7 times, we call it 7 transmembrane protein. And normally it can be a G-protein coupled receptor because this type of membrane receptor can bind to the G-protein. G-protein is a heterologous trimeric G-protein. You have G-alpha subunit, G-beta, and G-gamma. Okay? In the present, the GTP bound to this G-protein is in inactive form. But once this G-protein coupled receptor activated, it will trigger the exchange of the GTP with the GTP. Then it will become an active form. And once the G-protein binds to the GTP, it will dissociate this beta-gamma subunit. And then once beta-gamma subunit dissociated, the GS subunit become activated, and it can activate downstream factors, like, for example, adenylyl cyclase. However, normally, the GTP is unstable. It will be hydrolyzed, become back to the GTP. Okay? And once it becomes a GTP bound form, it will be able to we associate with beta gamma subunit and turn off the G protein activity. So, and that is how this activity, this signaling got, inactivated. And many type of the effectors are downstream of G protein coupled receptor. We already talked about the protein kinase A, right? Protein kinase B, and also this phospholipase A, blah, 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 and also the sodium hydrogen exchanger, and then actinic cyclase. And those things can have a transient cytosol transient response, or it can activate, for example, in this case, MEP kinase can go into the activating the MEP kinase cascade, go into nucleus and activate the gene repression or transcription. Okay, and then it can then have a permanent response by changing cell shape and others, or cell proliferation. Okay, then we're going to skip this one. Okay, and this is a slide for the cyclic AMP, and we already talked about that. Okay, then the cyclic is going to produce cyclic AMP, and the cyclic AMP can activate, let's say, protein kinase A. And the other very important second messenger, in addition to the cyclic AMP we talked about and cyclic GMP, is the IP3 and calcium. And actually, this IP3 and calcium can also be activated by activating the G protein coupled receptor. It has a G protein coupled receptor like there. Okay, it can activate a downstream signal called phospholipase C. Phospholipase C, by its name, it can clip the lipase activity. Lipase is that is enzyme actually can cut the membrane lipid. Okay, and the precursor for IP3 is a phospho 145 phospho45 bisphosphate, actually PIP2. Okay, and this PIP2, once it got cut by phospholipase, it can produce two molecules. One is the IP3, the other one is the DHG, which still reside on the membrane. Okay, and this IP3 is a well-known second messenger that can bind to the calcium channel on the ER and the plasmid reticulum. Okay, on the ER, there are many IP3 receptors. Okay, and once this IP3 binds this IP3 receptor, the channel can open. And once it's open, what's going on there? There's lots of really high concentration of calcium in the ER. Okay, in the ER. And the high concentration in the ER, once the IP3 receptor got open, it can release into the cytosol. So that means what? There's an increase in calcium concentration in the cytosol. Okay? And calcium is also itself a very important second messenger. Normally in a cell, the calcium concentration is a very low concentration. Okay, about 0.1 micromolar in the cell. Okay, 0.1 micromolar, that is a 100 nanomolar. Why we keep the calcium concentration in the cytosol so low? Because calcium is a killer. It can bind to the proteases. Okay, once the calcium binds to the proteases, it can activate the proteases. And proteases, by its name, is an enzyme can digest the protein. Okay, and once this protease digests the protein, actually protein degraded and cell will face the death signal. So that's why calcium actually normally keep it very low concentration at a resting state. But however, a lot of the responses, cellular responses, also rely on the calcium. For example, vesicle secretion. Okay, during the neurotransmitter exocytosis. You need high amount of calcium in order to trigger the exocytosis. So that means what? Sometimes we need calcium. Okay, and how can the calcium concentration get higher in the cytosol? That is controlled by this IP3 or other second messenger that can open the calcium signal, either from an inside store or from an outside store, because we also have a calcium channel on the plasma membrane. Once the calcium channel on the plasma membrane open, the extracellular calcium can goes into the cell. Okay, so there are two different ways, two different stores can contribute to the elevation of calcium. Okay, and in this case, intracellularly is caused by the IP3. And this IP3 is a signal can activate by G protein coupled receptor. But here, however, AP3 can also be produced by the other type of signal called tyrosine kinase. Okay, but I won't go into that today. Okay.
Excuse me, Jeff.
Yes.
Excuse me, Jeff. Apparently, Tsukuba side has some trouble, and
So we lost them.
Tsukuba side is not connected to.
Oh, fine.
Yes, yes,
Don't worry, we still have Kyoto
Yeah.
students. Okay.
Okay. Thanks.
Okay, so far is a basic description about the cell signaling. And for the last 10 minutes, I'm going to describe a little bit about the paper. Okay, give you a little bit about the background about the paper we're going to discuss presented by the students. And the paper actually is a publication done by one of my PhD students and published last year. And we use a zebrafish as a model, okay? And the reason we use a zebrafish because the zebrafish is embryo is very transparent, so we can observe the cell signaling in vivo in real time. So that's why we use zebrafish. This is an egg of the zebrafish embryo. And in addition to the transparency, the growth of the development of the zebrafish embryo is very fast. In 18 hours, it's pretty much like a small little fish. But normally, if you have a somatic cleavage for a cell, it takes at least 24, 1 to 2 days. And just within 1 day, you got a fish. And there are thousands of cells, okay? And in 3 days, 5 days, you pretty much got a free living larva. So that means what? All of the internal organ already developed. And they'll be able to move, they'll be able to eat, they're able to think, they'll be, swim. So it becomes a very, very good model to study early development and also the development of organs. Okay, and also is a vertebrate. So that's why it becomes a very important model for vertebrate development nowadays. And the story we're going to present, a student going to present to you is deal with the development of the zebrafish brain. Normally, a brain development is very, very complicated. And one of the key things for control the different region of development neurogenesis in the brain is the complementations. That's a subdivision of the brain, okay? And this is a figure about the zebrafish brain. You can subdivide it into forebrain and midbrain and hindbrain. Okay, and between the midbrain and hindbrain, students are going to talk about it. There's midbrain and hindbrain boundaries. Okay, right here. And normally, different components of the zebrafish can develop in a different pace. And the midbrain actually is the least the slowest one. Okay, and why it indicates in a slow development? Normally, you would need to have a more proliferating cell in order to generate more brain cell in that region. So somehow the zebrafish need to have a way to keep those midbrain cell as in differential state in order to trigger the development, get enough cell. And how can the differentiation of the developing cell got regulated? Normally, it's regulated by the control of the cell cycle. During early cleavage stages, the cleavage of the blastomeres actually is very fast. And the reason to do that, because normally in the cell cycle, we have M phase, G phase, S phase, and G2 phase. But during early cleavage stage, the G phase, growth phase, got eliminated. So the cell can be rapidly divided, okay, without the growing phase. But on the other side is because the cell becomes smaller and smaller because they never grow. Okay, but however, for the early embryos, the blastomere, the cell size is very big. Normally, our adult cell about 10 micron, and these cells can be up to 100 to 600 micron. So initially they have big size. So it doesn't matter; it has become smaller and smaller. Eventually, it goes into the adult cell. Okay. Then after they reach enough cell like this. Okay, this is 1,000 cells already. Gradually, the G phase adding back. Okay, you have G1 phase add back, you have G2 phase add back. And once it resume the normal cell cycle, those cells are ready to differentiate. Okay, so before they differentiate, they need to migrate. That is a very interesting issue. Okay, and once they migrate to the proper spot, the cell will in situ will start the differentiation. Okay, so once you have differentiation and then cell morphology will change, so you can specify into particular organ cell. And how do you know that? There's a change in cell cycle. Actually, you can have a cell cycle reporter. Okay, in this case, we use a fluorescent reporter called Fuji, fluorescence ubiquity machine-based cell cycle indicator. If it shows in red, that is the G phases. It shows in the green, it can either be S phases, G2 phases, or M phases. And this is a light zebrafish, okay? And you can see that injected with a Fuji reporter. And green, is less differentiated. Red become differentiated cell. This is muscle, okay. And you see here, two really region very remain as a precursor state, never differentiated. That is this spot called dorsal midbrain and also the retina, okay. And the subject for the paper will be study how this dorsal midbrain can remain undifferentiated during early development. Okay, so that will be an introduction for the paper. And I think we'll stop here. If you have no more questions, we can take a 5 minutes break and then we go to the student presentation. Okay? Okay, let's take a break.
everyone. My name is Albert Lan. Here are the speakers of today's presentation: Wang Ming, Chen Yun, Zheng Yazhi. We together are going to share this remarkable study. Stathmin-like 4 is critical for the maintenance of neuroprogenitor cells in dorsal midbrain of zebrafish larvae by Dr. Li Sijie and his then PhD student Lin Mengru. This is a very interesting research on how the neuroprogenitor cells in the dorsal midbrain are being maintained in an undifferentiated state during the brain development stage of zebrafish larvae. Brain development of the vertebrates has always been mysterious but intriguing. A fully functioning brain requires multiple brain regions to deal with their own task but work as a whole. Therefore, how brain develops into distinct brain regions through regulation of neuroprogenitor cells becomes the key question. Many proteins are involved in the maintenance of neuroprogenitor cells, and those proteins involved must be carefully expressed at a certain level. Either too high or too low would interfere with the normal process of development. Here we are going to show you an example, and this protein is stathmin-like 4, and we will call it STEM4 in the following presentation. Early brain development, including brain regions like telencephalon, diencephalon, these together form forebrain. Romobules behind the midbrain are also known as hindbrain in the later stages. Between midbrain and hindbrain, there is a distinct region called midbrain-hindbrain boundary, MHB. MHB is the crucial developmental signaling center responsible for patterning midbrain region of the vertebrate brain. This study focused on the midbrain, showing how STEM4 plays a part in the regulation of midbrain development. In molecular level, some well-known factors such as Wnt, fibroblast growth factor, FGF, play a huge role in the dorsal midbrain development. They are the commanders of these progenitor cells, telling where and when they should differentiate or stay undifferentiated. As you can see, this rectangular shape is the midbrain region of the zebrafish. A is for anterior, P is for posterior. The standing small rectangular area is MHB. When it is expressed in the MHB during the early stages of development, FGF activity gradient occurs throughout the midbrain. Expression of HER5 along the dorsal midbrain is dedicated by the FGF activity. HER5 acts to restrict neuronal differentiation of the neuroprogenitor cells. As development proceeds, FGF activity diminishes, resulting in a retraction of HER5 area. And this leads to neuronal differentiation, resulting in the formation of level 3 positive cells, neurons. In fact, HER5 prevents neuron differentiation through cell cycle regulation. Additionally, FGF activity can also be regulated by a negative feedback loop through splicing gene SPI, which is controlled by Wnt. This phenomenal regulatory system smartly guides the orientation of neuronal maturation, ensuring a well-functioning brain region afterwards. Since cell fate is regulated by cell cycle, we think of microtubule, a key component of mitotic spindles that drives cell division during cell cycle. The dynamic of those microtubule may be involved in the maintenance of neuroprogenitors. Several microtubule-associated proteins, including stathmin family phosphoproteins, mediate microtubule dynamic. Stathmin binds to tubulin subunits to form a heterodimer, sequestering tubulin subunit to polymerize. Once stathmin is phosphorylated, its binding to tubulin is inhibited. Dr. Li and his group have demonstrated that 4 out of 7 stathmin genes restricted expressed in the central nervous system during the development of zebrafish embryos back in 2014. SATHMIN4 showed its distinct expression within neural-free midbrain during the early brain development. However, other SATHMIN proteins can be observed in different regions of the brain. So they started to investigate STEM4 expression pattern during the midbrain development of zebrafish embryo by performing hormone in situ hybridization. The representative photographs are shown only after 11 hours post-fertilization because STEM4 was not expressed before then. STEM4 first appeared at the dorsal midbrain at 11 hours post-fertilization, and then were observed at ventral tegmentum till 20 hours post-fertilization. At 24 hours post-fertilization, you can see STEM4 dramatically decreased since neurogenesis of dorsal midbrain happens in the late embryogenesis. The author raised a hypothesis that STEM4 might inhibit neurogenesis during the early brain development. So they carry out double in situ hybridization against STEM4 and ELAV-like neuron-specific RNA binding protein, LVO3. LVO3 is a neuron-specific marker. The color purple indicates STEM4. Color red marks the LVO3, indicates neurons. Arrowheads points out the dorsal midbrain. As you can see, mature neurons first arouse at telencephalon and ventral tegmentum at 12 hours post-fertilization. Gradually expanded into trigeminal sensory neurons at 14 hours post-fertilization. Then epiphysis at 16 hours post-fertilization. Epiphysis is located within diencephalon, a part of the forebrain. But mature neurons were not observed in the dorsal midbrain region where STEM4 was expressed. The relationship between LVO3 and STEM4 is mutually exclusive. This supported the hypothesis that STEM4 could prohibit neuronal differentiation to maintain the neural progenitor cells in the dorsal midbrains. And now I'll let Wang Ming to continue the presentation.
Then they use two antisense translation blocking morpholino oligonucleotide, right as NO, to know whether STEM4 involved in maintenance the neural progenitor state in dorsal midbrain. Morpholino oligonucleotide is a molecular structure that blocks other molecules to small specific sequence of base pairing of RNA, about 25 base pairs. Which are used as research tool for knockdown gene function. In this paper, they designed two morpholino oligonucleotides. One target at start codon and another target at 5' UTR. To know the efficiency of morpholino, they co-inject PCS2 plus XLT GFP with an insertion of STEM4 containing both morpholino oligonucleotide target site with or without morpholino. And we can see in figure control without morpholino oligonucleotides express the GFP fluorescence. In contrast, there shows reduced fluorescence in morpholino inject embryos. Here we can see a percentage of embryo with green fluorescence compared with control. The percentage reduced in morpholino means the morpholino are efficient target and knock down the GFP expression as well as STEM4 function. To address the regulation of neurogenesis by STEM4, they use transgenic TG-HUC chiastic lines which expressing green chiastic fluorescence in different neurons. E indicator epiphysis and the TGSM means trigeminal ganglion sensory neuron. And dorsal midbrain located with them. between them. In control, the kaida level neuron meaning expressed in epiphysis and TGS and after 16 hours post-fertilization, and not found in dorsal midbrain. And in stage 4 movement, the four regions appear in dorsal midbrain, means the differentiated neuron appeared. And similar results were obtained by hormones in situ hybridization analysis against LVO3. From this data, we could learn the stem cell regulation regulate the neurogenesis. Here indicate the percentage of embryos with equivalent LVO3 expression in dorsal midbrain. The data shows LVO3 increased up to 80% of stem cell movement, both morpholino 1 and morpholino 2. The expression alleviated by conjugate with p53 morpholinos. Previous report, about 20% of morpholino used in Libra-free shows of targeting effect, which are mediated through the p53 induced apoptosis. So they used p53 knockdown as a tool to attenuate of targeting effect. And we can see there is not significant. And we can find morpholino 2 appear to be more effect than morpholino 1, so it will use for the rest of experiment. To figure out the precocious LVO3 expression observed in stem cell movement was specific due to the depletion of endogenesis stem cell. They co-inject with stem cell plasmid to embryos and found there is no significant effect on precocious LVO3 expression. The percentage increased after adding stem cell morpholino 2. And they also test whether the effect of stem cell movement might caused by non-specific activation of p53. Taken together, this result indicate the depletion of stem cell could specifically induce the precocious LVO3 expression in dorsal midbrain. To further confirm the function of stem cell genetically, they generate homologous stem cell mutant in transgenic TGSN HUC kaida background. Using the CRISPR-Cas9 mediated approach, they design several gRNA which target different sites in stem cell, then co-inject into one cell stage embryos. When F1 embryos grows 2 months old and subject to central sequencing analysis. And they found one of the gRNA target exon 3 sequence. We can show the sequence here. And they efficiently create deletion in 5 different mutant earlier. Like here, stem cell deletion 3, 4, 5, 7, and 12 of stem cell gene. To identify the homologous mutant, they use the BFA1 site, which in all the deletions and the second BFA site at the beginning of exon 5. By using restriction digestion assay to separate from wild type, heterologous and homologous mutant stem cell mutant fish. And we can take the stem cell deletion file as an example. The PCR product was cleaved by BFA1 into 3 fragments in sample from wild type embryos. In contrast, 2 fragments were present in sample from homologous mutant because they lost the first BFA1. And as heterozygous, we can find 495 and 371 base pair preset. So we can distinguish the homologous mutant from heterozygous and the wild type. They also found the two stem 4 mutant, stem 4 deletion 4 and deletion 5 are close to the pen site. Pen site is a short sequence immediately following the sequence targeted by Cas9 nucleus. Because they are near the pen site, causing the premature start codon. And result truncated stem 4 mutant of 28 and 36 missensing amino acid. This mutant lack all the stem 4 function domain. Then examine premature neurogenesis in dorsal midbrain in wild type and stem 4 mutant embryos. And exactly, most of the mutant embryos from those both earlier developed normally. Only 6.7% in stem 4 deletion 4 and 7.8% in stem 4 deletion 5 mutant embryos shows the premature LVO3 expression. They also test the response to stem 4 morpholino 2 and found the morpholino 2 induced premature LVO3 expression was significantly lower in stem 4 and stem 5, deletion 4 and deletion 5. This indicates that the observed premature LVO3 expression in dorsal midbrain was induced by stem 4 morpholino 2. They also observed lower but notably precocious LVO3 expression in dorsal midbrain in GRAS Cas9 treat embryos. This result suggests possibly functional compensation by other STEMI isoform or gene compensation that might mask the phenotype result from loss of stem 4. Then examine the gene expression of other STEMI gene in stem 4 deletion 5 mutant at 12 and 18 hours post-fertilization and found the expression of stem 1b, but not other gene, was significantly and notably elevated. To confirm the stem 4 morpholino-induced precocious LVO3 expression in dorsal midbrain, they use shRNA targeting the 3' UTR of stem 4 and employ the US amyl-13 backbone in combination with the GAL4 UAS system. One fact was contrast with the HISHA protein 17 promoter and the GAL4 gene. Another factor contained the UAS element and the TD tomato and the stem 4 shRNA, which will target nuclear 614 at 3' UTR of TD tomato and inject both the factor into one cell stage. And when culture to the bulb stage and then HISHA for 2 hours, the HISHA protein 17 promoter can activate by HISHA, then drive the GAL4 expression. GAL4 protein then bind to the UAS element and trigger the TD tomato and the stem 4 shRNA. By using the confocal microscope, the expression of stem 4 shRNA will express in red and LVO3 in green. And the asterisk means the cell expression for reasons above the certain threshold and was counted as a possible cell. A yellow one indicates a double positive cell. We can observe the stem 4 shRNA induced in induce the precocious LVO3 expression. We can observe the stem 4 shRNA. This result clearly demonstrates at the cellular level of that the loss of stem 4 induced precocious neural differentiate. They also test the effect of overexpressed wild-type stem 4 or constitutive activate stem 4. Observed in 3 trials, the overexpressed can induce a precocious LVO3 expression. Sure. From this data, we can get a short summary. We know the either loss or gain of stem 4 can induce precocious neural differentiates. So the stem 4 has an essential role in maintenance of neural progenitor cell.
To further investigate the function of stem4, they decide to overexpress CA stem4, which stands for constitutively activate STEMIN4 in vivo, and see what will happen in zebrafish embryo. They used the model which mentioned previously and add another vector medusa containing the double coating GFP to monitor the expression of microtubule. Double coating, which is abbreviated as DCX, is a microtubule-associated protein expressed by neuronal precursor cells. Neuronal precursor cells begin to express DCX while actively dividing, and their neuronal daughter cells continue to express DCX for 2 to 3 weeks as the cells mature into neurons. The neuron development within time are shown here. As first, they give a heat shock treatment at 6 soma stage, which neurons are still in neural plage stage. At about 13 hours post-fertilization, which is at neuron kill stage, they use time lapse to investigate the expression of tubulin and stem4 until 16 hours post-fertilization, and the neuronal kill stage have tangential neural rod. Here, author provide a short video of time lapse. The green fluorescence kaide presents cytosol, which represent the life cells, and the red fluorescence represent expression of stem4. As we know that red plus green will become yellow. So when stem4, which is red, show up in the same place as cytosol, which is green, the nucleus will become yellow. Therefore, the neuronal progenitor cells are shown in green cytosol and yellow in nucleus. It represents that it is a live cell which expresses stem4, and it will be indicated by an arrow in the video. If the green fluorescence are weak, as I draw here, this represents apoptosis cells, and it will be indicated by the arrowhead in the video. Between the two white dashed lines are the midbrain. The time of these two videos are synchronized, so we can investigate the difference within the two different treatments at the same time. In the video, we should notice that in the overexpressing CSTM4 treatment, there are more generation of neuronal progenitor cell and also apoptosis cells. Now, let's take a look at the video. As we can see in the CSTM4 overexpression treatment, both precocious and apoptosis occurred. Through time-lapse investigation, they also found out that approximately 71.4% cells underwent neuronal differentiation before cell division. Let's have a close look at the snapshots of the time-lapse result. DCX GFP still represents microtubule. DCX GFP still represents cytosol, and M-cherry represents the expression of stem4. The arrow represents the formation of neural progenitor cells. Otherwise, the arrowhead represents apoptosis cells. The upper line are control, and the bottom line are treatment which overexpress CS stem4. We know that time-lapse starts at 13 hours post-fertilization, which is a neuronal kill stage. And after 48 minutes, we can see a neural progenitor cell form in the treatment of overexpression of CA-STEM4. Within the time, there are more and more neuronal progenitor cells and also apoptosis cells occurred. Furthermore, some progenitor cells have morphological changes with fragmented microtubules and perform apoptosis regardless of ALV3 expression. This result indicates that overdose of CA-STEM4 can disrupt the viability through interruption of microtubule dynamics. And both precursors neurogenesis and apoptosis occur during the neuron kill stage in CA-STEM4 overexpressing embryos. So we should notice that both the deficiency and overexpression of CA-STEM4 will lead to the precocious neurogenesis. Then the author wants to know the mechanism underlying the early post-mitotic neuron cells appearance in the STEM4 movement. In order to investigate the length of M phase in embryos, they incubated BRDU, which is showed in green fluorescence, to label the S phase in the embryo, and injected pH3, incubate pH3, which is in red fluorescence, to indicate M phase. By calculating the time between the two fluorescence showed, we can get a G2 phase time duration. To understand the cell cycle duration, author incubated pH3 at different time in different embryos. For example, they incubate pH3 in one group of embryos at 17 hours post-fertilization, which is 1 hour later the incubation of BRDU. And we harvest the result at the first line, as we show here. At 18 hours post-fertilization, they incubated another group of embryos which were also incubated with BRDU at 16 hours post-fertilization and investigate the expression as we showed in middle line. Some as the third line which were incubated with pH3 at 19 hours post-fertilization. As we mentioned previously, red plus green will become yellow. In this case, S phase are shown in green and M phase are shown in red. Therefore, when a cell underwent S phase, G2 phase, and reached M phase, the color will turn to yellow. The upper row is the control treatment and bottom are added with STEM4 T-morpholina 2. First, we can take a look at the control treatment, and we can find that yellow signals are shown at 2 hours after the S phase. However, in the STEM4 knockout treatment, yellow signal showed lately 3 hours after S phase. In order to calculate the G2 time duration, author quantified the result. The horizontal axis represents the time after BRDU incubation, and the vertical axis represents the percentage of cells which express both pH3 and BRDU. As we can clearly see in this graph, the blue line indicates control, red line indicates STEM4 T-morpholina 2 with 5 nanogram and the purple one indicates STEM4 T-morpholina 2 with 10 nanogram. We set a standard at 50% of cells underwent S phase to M phase. We can see that control treatment have the shortest time duration from S phase to M phase. This indicates that control treatment had the shortest duration of G2 phase. Similarly, shortest at G2 phase. We set the standard at 50% cell underwent S phase to M phase, and we can see control treatment had the shortest time duration from S phase to M phase. This indicated that control treatment had the shortest duration of G2 phase. Similarly, the STEM4 knockdown treatment had relatively longer duration of G2 phase. It suggested that the control of G2M transition by STEM4 is a key to its maintenance of neuronal progenitor cells. To further investigate the relationship between STEM4 and cell cycle, authors chose to look at the CDC25A, which is an important protein at G2M checkpoint. They tried to overexpress CDC25A by introducing the vector of CDC25A into cells and investigate the expression of cells within STEM4 T morpholino 2. We can see in this graph, STEM4 T morpholino 2 used to have a high percentage of premature LFO3 expression. However, after introduced CDC25A vector into cells, the percentage of premature LFO3 expression decreased within the dose of CDC25A. It suggested that CDC25A might rescue the phenomenon of STEM4 T morpholino 2, and STEM4 might be the uppercase regulator of CDC25A. We know that STEM4 are related with cell cycle, so authors want to investigate the interaction between the STEM4 and another important cell cycle regulated protein, Wnt. LEF1 are a downstream gene which is Wnt activated, so it can indicate expression of Wnt signal. STEM4 are represented in blue dye in dorsal membrane in these pictures. Authors suppressed the activation of Wnt by applying a Wnt signal inhibitor. We can see that LEF1 was reduced within the time, showing that the activation of Wnt was successful. Then we can see that within the time, notable reduction of STEM4 expression is observed. These results are also shown by qPCR analysis in RNA expression of whole embryos in this graph. This indicated that Wnt signaling may regulate the STEM4 expression at a transcriptional level.
So after the experiment, here's some points we want to discuss. First, in this study, Sesame acts as a negative regulator for neurogenesis, and it appears to be contradictory to previous findings that sesamines are positive regulators for neuron differentiation. In those studies, STEM4 is expressed consistently at a high level during differentiation in multiple neuroblastoma cell lines, in spite of all described evidence disclosing the vital role of sesamine as a neural differentiation activator. The authors assert that STEM4 has a previous undescribed role as a neurogenesis blocker in developing zebrafish dorsal membrane. And the discrepancy may be due to the fact that mainly adult neurons or somatic cell lines were used in previous studies. Those cells are derived from terminally differentiated cells. In contrast, the authors used the developing brain in an in vivo context. In addition, what they based on is the expression of earliest neuron differentiation marker, LFO3, which represents as a much earlier signal compared to the previous morphological observation in neurite formation in culture. Therefore, the findings in this study may represent sesamine as a regulatory signal leading to the initiation of neurogenesis in dorsal midbrain. Next, when studying how STEM4 regulates the initiation of neurogenesis, some findings point out that there is a strong correlation between STEM4 expression and cellular proliferation. In previous studies, no matter in normal or malignant cells, both overexpression and inhibition of STEM4 expression give rise to mitotic arrest. This is not surprising since mitotic spindles, which are essential for chromosome aggregation and cell division, should be carefully mediated by microtubule-associated proteins such as sesamines. Since sesamines is crucial for mitosis progression, the lessening of G2 phase in STEM4 deficient embryos observed in this study might be due to the difficulty of forming mitotic spindle for entering mitosis. Moreover, the author observed that the STEM4 expression level in neuron progenitor cells should be properly controlled within a narrow window, that less or higher expression of STEM4 both resulting in premature differentiation in dorsal midbrain. As a result, it suggests that the homeostasis of STEM4 within neural progenitor cells should be strictly controlled for the maintenance of cell cycle progression. Then, about the role of STEM4 in cell cycle, here are some hypotheses in previous studies. They propose that histamine may stand as a gatekeeper at the G2M checkpoint via regulation of proteins which act upstream of CDC25A phosphatase to facilitate cell cycle progression. And this is in accord with those findings that CDC25A could rescue the precocious differentiation phenotype in STEM4 morphin. It supports to these studies that STEM4 controls the neuronal cell fate through mediating CDC25A, particularly during development. The cell cycle length hypothesis has been proposed and established to provide an explanation that cell cycle regulators could elicit effect on cell fate determination factors inversely. That is, retention of G2 phase line may allow accumulation of proneuron gene transcript to direct premature neuron differentiation, just like the regulation of G1 phase by other proteins. Constantly short G2 phase maintained by a proper level of STEM4 expression in dorsal midbrain to guarantee active mitosis in neural progenitor cell pools. In sum, the authors propose a model during the neural kill stage, while most cell cycle regulators participate in midbrain development situated in G1 phase, such as P27 and P57. Alternatively, STEM4 influence the cell cycle progression via regulating the G2 phase, which means we could regulate the expression of STEM4 in dorsal membrane to control the length of G2 phase, possibly via the coordination of G2M checkpoint machinery by activating CDC25A. In contrast, deficiency or excess of STEM4 would have a lower CDC25 activity, both lead to the retention of G2 phase that may allow accumulation of proneuron gene transcripts to direct premature neuron differentiation. At last, the conclusion of this study is that Wnt-mediated STEM4 homeostasis is crucial for preventing dorsal midbrain from premature differentiation via the G2 phase control during the neural kill stage. So that's all for our presentation. Thanks for your attention. Is there any questions?
So any question from our students? How about a Kyoto student? One from Kyoto side. Yeah, over there. Yeah, Kyoto student.
Hello. Thank you for the presentation. I just want to ask one quick question. I think you mentioned that the reason why they chose with STEM4 as a target is from previous studies, but I think from some of the data, I think from like when you mentioned like CRISPR-Cas9 knockout and like there's might be some compensation mechanism and whatnot. So did they mention or did they like investigate that further? Like did they like if the other stem, like I think just there's 1, 2, 3 or something, and so if like for example performing a double knockout or something and seeing if there's a significant change, or did they mention anything like that? I think yes, that part.
So we can see from this data that when we have a deletion, STEM4 deletion 5 knockouts, we can see STEM1B has a higher expression, relatively higher expression. But
So,
yeah.
But
You
they
mentioned about
didn't-
CRISPR-Cas9.
Yeah, like if I don't know, so I think they were suggesting that the other salmon, or like one, maybe in this like 1B might be important because there wasn't as much reduction, or so it might be like a compensation mechanism. So did they investigate that further? And
Okay, let me answer your question because it is our labs group's project. Okay, and basically based on the slide here right now, you can see the elevation of the specimen 1B at the later stages in the data for knockout. So one of the immediate question will be whether this compensation of elevation of the specimen 1B is a cause to mask the phenotype. So sure. I have one master student right now. He's going to work on this project, and actually he found that the specimen 1B did particularly elevated in the dorsal midbrain region. That is a really good hint that the elevation of specimen 1B in the dorsal midbrain region may interfere with the signal. So next he's going to do the double knockout to see whether the double knockout of specimen 1B and the specimen 4 can produce the precocious neurodifferentiation phenotype. But however, it takes a long time, and it doesn't guarantee that double knockout may have the signal. So one of the easiest way to do is that we can locally and conditionally inhibit the specimen 1B expression in the dorsal midbrain region of the specimen 4 knockout. So we are currently developing the contract to try to conditionally knock out specimen 1B in the dorsal midbrain region in a specimen 4 knockout using the CRISPR approach. And I wish we can get the data in a couple of months. Okay. If that turned out to be, if it turned out to be possible, that would be great. Now we give it more hope to generate double knockout to see whether that is the case. But however, knockout and knockdown, this kind of phenotype actually is really controversial recently in the zebrafish field, because we know that not only the zebrafish knockout, a lot of them do not have phenotype, especially generated by CRISPR, but also other species as well. You see, some of the knockout do not have the phenotype as well. So it's really, too subjective to say, if the phenotype of the knockdown and knockout are different, that your knockdown data will be not right. Okay, I think this is subjective. So we've got to be very cautious to interpret the data, and that is the thing we're currently working on it. Okay, I wish that answer your questions.
Okay, thank you.
Okay. Any more questions from the audience?
Hello, thank you for your presentation. So I was working with zebrafish once, although not transgenic. So I would like to ask whether this transgenic modification actually produced any visible change in the morphology or the behavior of the fish you got. So we have the heterogeneous, not heterogeneous, homogeneous and heterogeneous fish. So I wonder if there's any significant difference in morphology and behavior compared to wild type fish. Thank you.
Well, so far we have many mutant allele, but really the phenotype is really mild. So we cannot answer your question so far. But I think with our conditional knockout approach, we might be able to say something. Okay.
So it's actually this process of regulating differentiation in different stages do not produce any significant behavior change or something like that. So even if an individual have this, not only in zebrafish but in humans, they have this kind of dysregulation in the neuronal differentiation during development, then there would be no clinical significance to this research.
Yeah, I think that is one of the next project for us. We try to see whether there's a behavioral difference for this knockout, even though they do not have that kind of neurogenesis problem. And I think we have a new faculty coming back from the weekend, Japan, Green Science Center Institute. He's really good at the behavioral assay. So we are trying to collaborate with him to see whether we can see some behavioral difference there. Okay, that's a good suggestion though. Yeah.
Maybe a little something. Do you have any expectations what kind of behavior would change?
No, we do not have any behavioral data so far. No.
But maybe because it's at the boundary between midbrain and hindbrain, there's probably some maybe motor behavior or something like that. Because we know the function of the midbrain and the hindbrain, although they are quite complex. So maybe they would produce complex behavior, but probably using our previous knowledge about the functions of the midbrain and the hindbrain, maybe we can produce some hypothesis of what kind of abnormal behavior might come up.
Yeah, that would be great though. Yeah, thank you. Yeah. Well, other questions from the Kyoto side? Any from the NTU side? Well, if there's no more, I think it's 11 o'clock already here in Taiwan. So thank you so much for participating, and I wish to conclude the section now. Thank you so much.
Okay, thank you.
Thank you. Yeah.
By the way, so Tsukuba University, all the campus was down for the network, but they are now recovered. So probably next week should be fine.